This is Module 9, Fatty Acid Metabolism. In this video 9-2, Transporting Fatty Acids, I'm going to talk in great detail about the different lipoproteins that bind to fatty acids and tags and transport them to different destinations in the body via the exogenous pathway or the endogenous pathway, which were described briefly in the first video. Lipoproteins are specialized particles made up of fat droplets surrounded by a single layer of phospholipid molecules, thus creating something of a micelle. This micelle, so here we have our glycerophospholipids, which make up the outside of the lipoprotein. And so we have our polar heads in blue, and we have the hydrocarbon tails, the nonpolar tails, in the center. There are bits of cholesterol that are also inserted around this membrane that helps to maintain the fluidity and flexibility of the micelle. And then there are also triacylglycerols and cholesterol esters within the core of the lipoprotein. On the outside of the lipoprotein, there are special proteins called apolipoproteins, which coat the surface and are specific for each type of lipoprotein. So it helps to serve as ligands for receptors on the outside of cells that can absorb these lipoproteins and internalize fatty acids. And so the lipoprotein is this entire molecule. The apolipoprotein are these smaller ones that are embedded within it. And they help to determine the fate of where this thing is going. So it can bind to specific receptors and become internalized, say on the liver or muscle tissue. And that's how it helps. That's how we specify the destination of some of these fatty acids, is through the identity of these apolipoproteins. And so fatty acids really enjoy being in the core here because the core is made up of nonpolar phospholipid tails. And so because they are surrounded by these polar heads, they can transport easily through the bloodstream without clustering together and causing problems. So there are different types of lipoproteins: chylomicrons, very low density lipoproteins; intermediate density lipoproteins; low density lipoproteins, that probably looks familiar to you; and high density lipoproteins, this also probably looks familiar to you. These are your bad and good cholesterol, that which is like sort of the layman's term for referring to these types of proteins. The lipoproteins are characterized based on their density. So the very low density, sorry, this is an increase in density. So very, very high dense lipoproteins are characterized as a high concentration of protein and not a whole lot of lipid. And so these chylomicrons represent the least dense lipoprotein because they contain the most amount of lipids. These are the ones that help to absorb dietary fats. The rest of these, so the VLDLs, IDLs, and LDLs, are ways to transport adipose tissue stored fats throughout the body, and they vary in protein concentration and lipid concentration. It starts out as a VLDL, and so as lipids are cleaved off of these lipoproteins by tissues that use fatty acids, that will decrease the amount of lipids in the lipoprotein and therefore increase the density of the lipoprotein. And then high density lipoproteins serve a different function. They're not really involved in either the endogenous or exogenous pathway of fatty acid mobilization. They're more involved in quality control. And so I'll talk about that towards the end of this video. So as I mentioned, lipoproteins are transport molecules. And they help to facilitate transport of lipids via the exogenous pathway, or the transportation of dietary lipids, or the endogenous pathway, the transportation of stored fatty acids from adipose tissue. So the chylomicron is the lipoprotein that transports dietary fat into tissue or to the liver. And so this takes fat absorbed from the intestinal lining, packages it with cholesterol to form a chylomicron, and then either delivers it to tissue that can use fatty acids or delivers it to the liver. The liver can actually produce fatty acids using excess carbohydrates and cholesterol, And so excess carbohydrates are turned into triacylglycerols and then mixed with cholesterol to form this type of lipoprotein called VLDL, or a low density lipoprotein. This helps to deliver endogenous fatty acids and cholesterol to muscles and tissues that can use it. And so the liver is once again involved in a very important metabolic process, which is allowing the transport of endogenous fatty acids that are produced by the liver. So let's take a look at the exogenous pathway and how it relates to lipoproteins. Dietary tags are absorbed through the intestinal lining and packaged with cholesterol inside an intestinal epithelial cell. And then when this occurs, when you mix tags with cholesterol and phospholipids, you get a chylomicron. And so we absorb fat through the intestine, and within an intestinal cell, chylomicrons are produced. Chylomicrons will either move towards tissue or move to the liver. And so when it moves to the tissue in the capillaries, once it gets to the capillary level, there are lipases, enzymes that break up fatty acids, in the capillaries that can bind specifically to chylomicron lipoproteins, break off tags, and produce fatty acids and glycerol. And then in the muscle, fatty acids are oxidized for energy. If the chylomicron makes it to the adipose tissue, fatty acids are they're rebuilt as tags for storage. Chylomicrons, so then what you have left over after all those fatty acids are removed are chylomicron remnants, which are exported to the liver. And then, it sort of recycles the apolipoproteins on the surface and the cholesterol embedded within the membrane so that, waste not. The body doesn't like to waste. Chylomicrons can also be delivered directly to the liver if there's an excess amount of, for processing by the liver. So chylomicrons can be destined for tissues or the liver. And tissues include muscle, adipose tissue, and also maybe the heart, which can also use fatty acids as a source of energy. Okay, so now let's talk about VLDLs, which is the lipoprotein that begins the endogenous pathway of lipoprotein metabolism. So once chylomicron remnants are brought to the liver, which is, lipoproteins and cholesterols, and also fat is delivered to the liver, the liver will synthesize particles called VLDLs, which are transported to tissues and metabolized by lipoprotein lipases in the capillaries so that tissues can use them as a source of energy. So this is the endogenous pathway. Fats that originate in the liver that are produced using excess carbohydrates as a source will be packaged with VLDLs, or fatty acids that are brought to the liver after they're mobilized in the adipose tissue. So the liver can repackage those fatty acids into VLDLs, very low density lipoproteins. They will travel to tissues and be metabolized. And so a few fatty acids will be cleaved off of VLDLs, which is going to, once again, reduce the lipid content and increase their density. And so VLDLs, when enough fatty acids are cleaved off, will turn into IDLs, or intermediate density lipoproteins. These intermediate density lipoproteins are taken up by the liver. About half of them are taken up by the liver, and then the rest are converted into LDLs as more fatty acids are cleaved off of them by lipases in the capillaries. And so that will create an LDL. And so the liver is very good at binding IDLs. And so when this happens, you remove fat from the bloodstream, the liver repackages it, and then we repeat this process. But again, only half of them are able to do that. The other half are converted into LDLs. And LDLs can sometimes circulate in the bloodstream for a very long period of time before they are taken out of the bloodstream. So I'll talk about that in a moment. I'll come back to this idea of bad cholesterol or LDLs. So let's summarize the pathway. So once again, chylomicrons mediate the exogenous pathway, fat absorbed from the intestine, or fat that, fat absorbed from the intestine will move to the liver. And so that is beginning, we're here, the exogenous pathway. So dietary tags, phospholipids, and cholesterol are absorbed into the intestinal lining. Some fatty acids are cleaved off by tissues that use them as a source of energy, and then they make their way to the liver for uptake. So chylomicrons bind specifically to a receptor that recognizes apolipoproteins on the outside of the chylomicron. And if this receptor is broken, this is the disorder that results. So that's a genetic disorder. VLDLs, IDLs, and LDLs mediate the endogenous pathway, where fat from the liver goes to peripheral tissues, or so they use them as a source of energy, or to adipose tissue for storage for later. And so that's this pathway here. So we start in the liver when we're talking about the endogenous pathway. We've got fats mixing with cholesterol, a few phospholipids, and apolipoproteins, and then we create the VLDL. Fatty acids are cleaved off as VLDLs bind to specific tissues that can use fatty acids as a source of energy, creating IDLs. Half of those are taken back up by the liver, and then we repeat this process, this circular process here. The other half will undergo more fatty acid cleavage to produce an LDL, and then some of those are taken up by the liver, although not as easily as IDLs. So LDLs are not absorbed by the liver as easily, and so they tend to sort of circulate throughout the bloodstream. If LDLs circulate in the bloodstream for too long, you end up with an immune response where macrophages will come try to clear them out of the bloodstream, recognizing them as something dangerous, which can sometimes lead to the production of a plaque. And so this is why LDLs are really not great for your body. If they circulate too long in the bloodstream, they induce an immune response, which causes plaque formation and sometimes can lead to heart attack or stroke. HDLs are a separate type of apolipoprotein that handles reverse transport. And so HDLs are made by the liver and put into the bloodstream. And HDLs are responsible for picking up other lipoproteins, cholesterol that's left over in the bloodstream, and so sort of cleans up the mess left behind by excess LDLs. Let's take a closer look at the HDL. This is your good cholesterol. And so your doctor will tell you high levels of HDL are associated with a low risk of heart disease. High levels of LDL are associated with a high risk of heart disease. And so what they like to look at is the ratio of HDLs to LDLs. If you have a good amount of LDLs, but you have a greater amount of HDLs, you're not at risk quite yet because your high HDL lipoprotein amount is helping to scavenge and pick up some of those LDLs. The role of an HDL is this scavenging, and so it helps to deliver cholesterol to tissues that need it or to other lipoproteins. It also helps to pick up cholesterol in the tissues and bloodstream and bring it back to the liver. And it does so based on its specific apolipoprotein identities. So here's the apolipoprotein identifiers on the outside of these HDLs. There's E, which are found in chylomicrons and IDLs. And so when HDLs bump into chylomicrons and IDLs, it will donate the ApoE apolipoprotein. Okay. ApoE is really good at binding to the liver. And so the importance of HDL is it's able to donate ApoE to IDLs that are migrating around in the bloodstream, which helps to the liver bind to IDLs and pull them out of the bloodstream so it doesn't clog up the bloodstream. ApoEs also are found in chylomicrons, which again helps the liver bind to chylomicron and internalize it. So this ApoE protein is a liver-specific protein. It recognizes a receptor on the liver, binds it, and it's internalized. And so HDLs help to deliver these apoproteins to other lipoproteins. ApoA1 is found in most HDL proteins. It allows the transport of cholesterol to the liver, back to the liver, to get cholesterol out of the bloodstream, to tissues that need to use it, or other apolipoproteins that need to be built. ApoC2 is found typically on chylomicrons and VLDLs, and it's responsible for activating the capillary lipases, the lipoprotein lipases, that help to cleave fatty acids off and allow for tissues to oxidize them for energy. And so HDLs have all of these apolipoproteins. And so one of the roles of an HDL is to bind or to bump into these other particles and donate apolipoproteins. And that helps the liver pull them out of the bloodstream, especially if it's donating ApoE. It also helps chylomicrons and VLDLs bind to their target tissues. And so HDL is really important for helping the other lipoproteins bind to their targets and getting them out of the bloodstream. So new VLDL particles will bump into the HDLs, which donates the appropriate apolipoproteins to it, which will make it mature. Okay, so let's take a look at this. So here now we have our lipoproteins. We have our chylomicrons, our IDLs, our LDLs, VLDLs, so all of the lipoproteins with the exception of HDL. And so chylomicrons have these two apolipoproteins, C2 and B48, that helps it bind to target tissues. So again, ApoC2 helps to activate lipase enzymes in capillaries, which will break off free fatty acids and allow tissues that need them to utilize them. So this protein helps to activate fatty acid cleavage so that tissues can use fatty acids. IDLs have ApoE and ApoB100, and ApoE binds really well to the liver. And so when, oh, wait a minute. I think I didn't really start where I should have started. Okay, sorry. So here's the exogenous pathway. ApoC2 and ApoB48 bind to chylomicrons. Here's the endogenous pathway, starting with VLDLs, which also have ApoC2. So VLDLs can deliver fatty acids to tissues. They're cleaved off. And when this occurs, during the transformation of VLDL to IDL, we lose the ApoC2 and it's replaced with ApoE. And HDL is what gives IDL its ApoE. Now it can bind to the liver and be taken up out of the bloodstream. If IDL is further cleaved, and so more fatty acids are pulled off of it, before it has a chance to get to the liver, it's turned into an LDL. And the problem with LDL is it loses this ApoE binding site. And so this is why IDLs can be taken up by the liver and removed from the bloodstream, but LDLs have a much harder time. ApoB100 doesn't bind to the liver as easily, and so it's not as absorbed out of the bloodstream. And this is why LDLs tend to circulate more in the bloodstream. So again, as VLDL particles are metabolized, they become more dense. And ApoC2 will activate lipases, allowing them to be metabolized, and then we convert VLDL to IDL. VLDL will swap its ApoC2 for ApoE during this process, and that allows IDL to become bound to the liver and taken out of the bloodstream. However, on its way to the liver, some lipases will still pull fatty acids off of this, turning it into an LDL, and that's also going to make it lose its ApoE. And so now the LDL cannot bind to the liver and will circulate in the bloodstream. Okay. Some LDL, so again, LDL doesn't bind as well, but it still binds to the liver. And so if there's a low amount of LDL, if you have a low amount of bad cholesterol, your liver can pretty much take care of that circulating LDL. And your high HDL level will also donate apolipoproteins to LDL, making it easier for the liver to internalize it. And so as long as you have low amounts of LDL, your liver can probably keep up with the pace. As long as you have high amounts of HDL, that will donate apolipoproteins. That means the LDLs can be taken up by the liver even better. However, sometimes you can have too much LDL. So when your bad cholesterol is too high, that means your liver is not going to keep up with the pace of how much LDL is circulating in your blood. And also, if your HDL is low, it's not going to be able to help out and scavenge the bloodstream for these bad guys. And so that's when you end up with high cholesterol and a risk of heart disease. So how does this work? Here we have our LDL receptors. LDL is internalized when it binds to these receptors. So it binds to these receptors, they cluster together and then engulf the LDL protein. The free cholesterol and fatty acids are released into the cytosol as lipases will release them. And so within this vesicle, it transports to the lysosome where all those nice hydrolytic enzymes are. And the lysosome will break up cholesterol esters, will break up ester bonds, tags, and all that. And so it's going to release, also there's proteases in the lysosome too. So that's going to break up any proteins into amino acids, break up your cholesterol, break up your fatty acids. The LDL receptors are then put back on the surface. And so if they're so this helps the liver recycle cholesterol. Your liver can also make cholesterol, but if it's getting enough scavenged cholesterol from the bloodstream, it will not make as much because it's being able to recycle stuff from the bloodstream. And so a high cholesterol concentration in the ER, the endoplasmic reticulum, will decrease the rate of cholesterol biosynthesis. And so if you have a lot of cholesterol already, the whole purpose of internalizing an LDL is so the liver can use cholesterol that's circulating instead of building it itself. And so it will not build its own cholesterol if it's able to get cholesterol from an outside source. If it's able to internalize that cholesterol and also the rate of biosynthesis goes down, this means the liver views this as, well, I don't need any more cholesterol because I've already got plenty of it to use to make what I need to make, cholesterol-based hormones, bile salts, all that stuff. And so what happens in response is a reduction of the LDL receptors on the outside. Don't bother making these receptors. I've got plenty of cholesterol already. And so if you are consuming more cholesterol, this means there's fewer receptors on the outside of the liver, and so less of that LDL is internalized. And so this is how consumption of too much cholesterol can reduce your LDL receptors, since your liver already has all the cholesterol it needs and doesn't see the point of using more of it, taking up more of it. So LDL is really bad because it has a harder time binding to the liver, and it can accumulate in the blood vessels, which induces an immune response and the formation of plaques. So LDLs have that ApoB100, but is lacking that ApoE, and so it doesn't really bind very well to the receptors on the outside of the liver without both of those present. Remember that IDLs do have both of these, and so it does bind very well to the liver. And so LDLs tend to circulate in the blood plasma for about 2 to 4 days, accumulates on blood vessel walls, induces an immune response, and causes heart attacks or stroke. Your HDL lipoproteins, or your good cholesterol, helps to extract excess cholesterol from the cells. So it scavenges for cholesterol and brings them back to the liver, and it's very good at binding to the liver. It also helps to donate apolipoproteins to other lipoproteins circulating to help it bind to either tissues very well or the liver very well. So it helps to scavenge not just cholesterol, but also helps other lipoproteins bind to tissues and internalize into the bloodstream, get out of the bloodstream. So because HDLs help LDLs and other lipoproteins bind to either the liver or tissues, there is an inverse relationship between HDLs and the amount of circulating cholesterol and therefore cardiovascular disease. And so here's this same pathway again that I introduced in the first video. The exogenous pathway, where we consume dietary fat, it's broken up in the stomach and then again in the large intestine, mixed with bile salts to create micelles, and then cleaved by pancreatic lipases into free fatty acids. Those are absorbed through the intestinal cell, repackaged into tags, packaged with chylomicrons, and exported into the capillaries and eventually to the blood vessels. In the capillaries, they can be cleaved by lipases, and so those free fatty acids will be absorbed by either the adipose tissue for storage or cells for oxidation and generation of ATP. And then they can also, those remnants left over from after you cleave off all those fatty acids are bound by the liver receptors and taken out of the liver. So again, we can get rid of those cholesterol pieces that are left over and put them back in the liver. The liver will then sometimes build fatty acids using excess carbohydrates as a source, and then package cholesterol with fatty acids into VLDLs. VLDLs will get into the bloodstream and be cleaved by lipases that recognizes the ApoC2 lipoprotein. And so that will be absorbed by adipose tissue for storage or be used by cells for oxidation and the production of energy. Cleaving fatty acids off of VLDL generates an IDL, which now has ApoE on its surface. And so ApoE will bind to the liver and be internalized, where we can recycle some of that cholesterol, rebuild VLDLs, or whatever it wants to use that cholesterol for. Some IDLs, however, experience further lipase cleavage, where fatty acids are cleaved off to produce LDLs. Remember, it loses its ApoE at this point, and so some is bound to the liver and can get into the liver, but a lot of times it can be circulating through the bloodstream for several days. HDLs are scavenging the bloodstream looking for lipoproteins to bump into and donate different apolipoproteins. So it's going to tag maybe LDL with an ApoE so that it's easier to bind to the liver, or it's going to donate some apolipoproteins to VLDL, which will mature them. It also will scavenge and look for any cholesterol within the bloodstream and bring it back to the liver. And so the more cholesterol is brought to the liver, the less likely the liver will make its own cholesterol. And if the liver has an excess of cholesterol, it will downregulate the amount of receptors on the outside so as to prevent any further uptake of cholesterol. If you're consuming too much cholesterol, that means it will then just be committed to circulating through the bloodstream, which causes problems for heart disease.